In this video, we'll be talking about the topic of mutations. A mutation is any change or mistake that occurs during meiosis to chromosomes or to any DNA sequence. There are two major types of mutations. We have germ cell mutations, which we also know as gametes. These type of mutations occur in gametes, therefore they are able to be passed down to offspring. These are the most harmful type because they can affect the next generation. We'll be talking about certain types of inheritances with germ cell mutations later on. The other type of mutations are called somatic mutations. As we all know, somatic cells are body cells, and these are mutations that happen in body cells, and because they don't get passed down to the next generation, these are mutations that stay with that particular individual and they do not get passed down to anyone else in the next generation. They just stay with that particular individual. So these are less harmful. Obviously, they're harmful to the individual themselves, but the germ cell mutations that occur in the gametes are way more harmful because they can affect more than one individual in the particular family. Now, normally when we think of the term mutation, we often don't think of it as being a positive thing, which oftentimes they're really not. Most mutations are either harmful or lethal or deadly, which obviously means lethal. But some of them can be beneficial. Some of them also can go unnoticed. There's a certain type of mutation that's called a silent mutation, where we're going to be talking about that in just a little bit, but they don't have any effect on the body whatsoever. They're a mistake that doesn't cause any consequences. But there are certain beneficial mutations. One mutation, as an example, that could be beneficial would be if you are a carrier for sickle cell anemia, you cannot get malaria. There's a particular gene mutation for anyone who is a carrier, so that this person not only does not have sickle cell, they're just a carrier, which means that they only carry one gene for it, which they can pass down. But if they are a carrier for sickle cell, they cannot get malaria. There's something with the shape of the blood cells, that doesn't allow that carrier individual to get malaria, which obviously would be a good thing if you were traveling in an area where you could possibly get malaria. Like I said, that's just an example. There are other beneficial mutations, but like I said before, most of them are harmful, especially, to the point where they could become lethal. Chromosomal mutations are extremely harmful because they're affecting multiple genes at one time. Chromosomal mutations typically occur in meiosis, and they result in a process of non-disjunction, which just simply means that there's an uneven splitting during either meiosis 1 or meiosis 2. So if you take a look here at this image that we have, this would be our homologous chromosome pair. Let's just say this comes from mom and this comes from dad. One whole chromosome should be going to one cell. The other whole chromosome should be going to the other cell, but because non-disjunction has occurred, you may have one of the full chromosomes going one way and half of the other homologous chromosome going the other way, which would result in one cell being called trisomy, tri meaning 3, you're having 1, 2, 3 copies of the chromosome in one cell, and the other cell would end up with monosomy, which mono meaning 1, you have one copy of that chromosome in that particular cell. So typically what happens, if this continues, let's just say that these are egg cells or these are sperm cells, it really doesn't matter which one. If they are chosen during fertilization, the trisomy, depending on what pair of homologous chromosomes were happened to be added in, let's just say if it was chromosome group 21, then that would be Down syndrome. If it's chromosome group 18, that would be Edwards syndrome. So depending on, which actual pair is the one that goes through non-disjunction would depend on the different the different disorder that baby would get. Now, monosomy, if this is the cell that's happened to be chosen for fertilization, that would most likely result in a miscarriage because there's just not enough genetic information to successfully, have a baby grow. So like I said, trisomy could result in various different genetic disorders. Like I said, Edwards syndrome is an example, Down syndrome is another. Monosomy, if chosen during fertilization, would most likely result in a miscarriage. Again, chromosomal mutations overall are the most harmful because there are many, many genes on one chromosome. When you start manipulating chromosomes, you're manipulating tons of genes along with that. You're not just changing or mutating one gene. There are many genes that are carried on that chromosome. So here are 3 examples of chromosomal mutations that can occur during non-disjunction. We could have a deletion, and that would be the deleting of one whole portion of a chromosome. So if you see here, this yellow piece would be removed during some portion of meiosis, obviously resulting in a loss of certain genes and causing a certain type of mutation. Again, I can't tell you what specific kind of mutation because depending on which exact chromosome, which exact genes are affected, would depend on the disorder itself. Inversion is where you have a certain segment of the chromosome. So if you note here would be a set of genes in red and a set of genes in yellow, and they flip within the chromosome, so now the genes are out of order. So that's inversion. And translocation is where you have a mix and a match, a deletion and an insertion or an addition of 2 non-homologous chromosomes. So if you notice here, we have our homologous pair here that is yellow and our homologous pair here that is blue, and you have this little red piece right here which is going to become deleted from one set of homologous chromosomes and added down here to completely non-homologous chromosome. So we know that this chromosome here and this chromosome here are not carrying the same genes. So not only do you have a deletion, but you have an addition of something that didn't belong in the first place. Translocations, mutations are very harmful because they're affecting multiple genes. They're all harmful because they're all chromosomal mutations, but out of the 3 of them, translocation would definitely be the most harmful. Again, depending on how large the chunk of the deletion is, depending on how inverted or how many times it inverts. These are all very subjective, but translocation, is taking non-homologous chromosomes and mixing and matching genes that don't even belong on, that aren't even linked. Now let's move into gene mutation. So now we're going to zoom in into the chromosomes and look specifically at the DNA sequences. These mutations, although not as harmful because you're not affecting as many genes, still can cause many genetic disorders and still can be very harmful or even lethal. So when we talk about gene mutations, there are 2 major types that we can talk about. There are point mutations, which is what we'll go over right here, and then we also have another kind which are called frameshift. So what I mean by a point mutation is that one specific letter in a codon is affected. So the way that can happen is you can either have a substitution where one nucleotide is substituted for another. So let's just say if you're supposed to have ACT as a codon, maybe the A gets substituted for a C, so then your new codon is CCT. You're just substituting one of the nucleotides. You could also have deletion, and deletion would result in a frameshift because what you're doing is if you take out one letter, all of the other letters are going to shift over and change all of the what we call frames or codons, which would change every other nucleotide sequence in that sequence. The other would be an insertion, which is also another type of frameshift. Difference is you're going to be inserting in a nucleotide, shifting the frame the opposite direction. So there are many types of substitution mutations. I know I mentioned the silent mutation before. These are the best kind of mutations because there is no consequence. It really is just luck of the draw. You do have a substitution mutation, but you get lucky and the amino acid ends up being the same. So let's take a look at the example that we have here. Let's just say the code is supposed to be GUU, which if we look in our codon wheel is valine. And let's just say there was a substitution for that last U to be a C. If we take a look at our codon wheel, we notice that GUC is also valine. So even though there was a substitution mutation, you just got really lucky and it just happened to be that that mutation coded for the same exact amino acid. So that is a silent mutation. These go unnoticed all the time. Unfortunately, you don't know when they would happen. They could happen all the time and you never know it. But then again, it's really just the luck of the draw. If this happened to be a different letter that would code for a different amino acid, then you would have a mutation that would be harmful in some way. Moving on to a missense mutation, which is very similar to the silent mutation, except for you don't get so lucky and you are coding, unfortunately, for an amino acid that's not supposed to be made. So let's take the GUU again, which is valine, and let's just say that the second U codes for an A instead. GAU does not code for valine, it codes for aspartic acid. So in that amino acid chain, you have now changed the shape and the structure of that protein. You are now making a completely different protein, which is a mutation. And again, depending on what protein this valine was supposed to be part of, and now is an aspartic acid, it is going to be non-functioning and it would cause some type of harm. So this is a harmful type of mutation. Moving on to, let's see what we have here. Okay, so moving on to a substitution. So if we have our DNA sequence here, I've blocked out the codons for you. Let's just say that we have this G substituting this C. The C would leave, the G would come in. The methionine would now not be made. Instead, we now would have isoleucine. So that's just an example of what a substitution mutation, so this would be a missense mutation because instead of producing methionine, you are now producing isoleucine, which changes the amino acid chain from methionine, arginine, threonine to isoleucine, arginine, and threonine, which is actually even worse because methionine is the start codon. Now this actually wouldn't even be produced because there is no methionine to get that started. So this one is actually even more harmful than normal. Let's take a look now at our frameshift mutations. These are our insertion and deletions. So as I stated before, one single nucleotide base is either inserted or deleted. Depending on that, your result is going to be shifted over. So just using the example of these three-letter words being your codons, the fat cat ate the rat, if the H is deleted, that shifts over every other letter to the left, and now you have an unreadable sentence, which therefore, take that analogy and put that into the way that amino acids are read and how the mRNA and the tRNA, how they read that, it won't make sense, and that's going to make for an unusable protein. All right, so here we have an example of an insertion. I'll actually redo that. So if we have our DNA strand over here, we want TAC. Over here in the mRNA, we want AUG. We want this to code for methionine. This is an example of an insertion where the T is inserted here right after the A, right before the C. So if you notice, now everything has shifted down. Now my first codon is TAT, which will change my mRNA to AUA. That shifts all of these frames. That changes methionine to isoleucine. Now we have alanine, and now we have tyrosine. That's changed 3, and obviously would continue more because this would be a longer chain. This changes all of the amino acid that corresponds to this protein, therefore resulting in a much more harmful and much more complicated mutation. So just to go over all the different types of mutations, if you're using this top one up here as your template, this would be the strand that we would want to use. So this is going to be our comparison. So we would want the DNA to read ATG as the first codon, CCA as the second, TCG as the third. We want methionine, proline, and serine. So if you look here and compare the silent mutation, this T here is going to be changed, right? So that A turns into a T, but CCA and CCT both produce proline, so therefore your amino acid sequence hasn't changed, and therefore there is no foreseeable mutation, but we all would know here that that is a mutation. That's why it's called silent. The missense mutation, same thing. Let's say this C turns into an A. That changes the proline. Okay, so now we have a different amino acid sequence here. So that is, but that's also only the change of one amino acid. If we look here at our insertion, our frameshift insertion, remember frameshift could either be frameshift deletion or frameshift insertion. Again, shifting it either to the right or the left. If it's a deletion, it shifts it to the left. If it's an insertion, it shifts it to the right if you're reading it like this. So here we would have a G inserted after the T. I'm sorry, the G after the G. And then you would be changing, so you have methionine here, it starts here after the proline, everything gets shifted over, and now everything after the methionine would be different. Sometimes this causes the stop to be changed, so remember if there's no stop codon, then this will continue to be read, so that can also cause an excess in amino acids and you know, to the point where the amino acid just keeps growing and growing and growing without stopping because the stop codon has been mutated as well. So that's another thing that could happen with that.